Good morning, everyone. Welcome to the third day session of Faculty Development Program. Myself, Dr. K. Swati Priya, co-convener of Faculty Development Program conducted by Srinivasarao College of Pharmacy on the theme Challenges and Innovations in Advanced Pharmacognosy. Our today's topic is Digital Pharmacognosy. Now, I request Mr. M. Ganesh Kumar, HOD of Pharmacy Practice, to introduce our today's resource person.
Good morning, everyone. This is M. Ganesh Kumar from the Department of Pharmacy Practice, Srinivasarao College of Pharmacy. It's my privilege to introduce our today's resources person, Dr. Satya Obul Reddy ma'am. Dr. Satya madam completed her B Pharmacy from Sri Vishnu College of Pharmacy in Bhimavaram and M Pharmacy in 2011 from Andhra University and PhD in 2023 from Andhra University. She served as assistant professor at Aditya College of Pharmacy, Sarampet, and she served as guest faculty in Andhra University AU College of Pharmaceutical Sciences. Currently, she working as professor at Pratiksha Institute of Pharmaceutical Sciences, Guwahati, Assam. Dr. Satya has published many research papers in various national international journals and written 3 book chapters. Dr. Satya has participated and presented papers in many international and national level conferences, seminars, and faculty development programs. Dr. Satya is EPTI lifetime member. She area interests are phytomedicines, nanoparticles, and nutraceuticals. Ma'am, it is a privilege to have our section today. Thank you for accepting our invitation, ma'am.
Thank you very much, sir. Good morning, everyone. It's my privilege and it's my honor to be here for the third day FDP session on challenges and innovations in pharmacognosy and phytochemistry. And I thank each and everyone for inviting me as a resource person for the third day FDP. Thank you very much. So today I'm going to start my session with traditional pharmacognosy and the digitalization in pharmacognosy. So, pharmacognosy, as we all know, it is a branch of bioscience. So, we all know the definition of pharmacognosy from C.K. Kokate textbook, Trees and Divans, and other various textbooks. Like it's a branch of bioscience which deals in detail with the medicinal and related products of crude and primary type obtained from the natural sources, which do possess some therapeutical action. But before the evolution of this exact definition of pharmacognosy, it was originally evaluated as a meta-discipline in the stream of pharmaceutical sciences. So meta-discipline in the sense this is the core subject from where we are having till the formulation and evaluation, like the phases of clinical trials and drug discovery, molecules and everything. So here, the basic and the initial definition of pharmacognosy is it was initially defined as the identification of novel drug molecules from biomedicine, which has toxic principles. Now, if we go to the definition of whatever the secondary metabolites it is, if we go to the definition of secondary metabolites, we'll be studying as they will be showing activity when taken in small doses. Now, if the dose has been exceeded, this results in the toxicity. So, the dose of the drug which we consume inside for showing any therapeutical property, this will be showing some activity to treat some diseases. But if this dose has been exceeded, or else if this dose has not been cleared properly from our body, this results in the toxicity. So that toxicity starts from hepatotoxicity to the renal toxicity. Now, here the natural biomedicine, the active principles of natural phytocomponents, these have been possess lot many uses, starting from a simple body pain to treat the viral infections or the cancer causing agents, etc. Now, if we go to the definition of pharmacognosy, it is gradually converted from the use of toxic principles till the use as a therapeutical agent. Now, during this conversion in the pharmacognosy, the database which we have used till the time of evaluation of the exact definition of pharmacognosy is just our Materia Medica and Papyrus Ebers. So these two are the books which we have got from the ancestors. The knowledge has been documented and encrypted in these two books only. Now we have the data, but what about the mechanism of action of the particular phytocomponent and all? Now, scientists, they have been advanced in the research arena regarding the phytocomponents, how the molecule has been formed, that is the biochemical analysis, biochemical pathways. Now, after elucidation and isolation of a particular phytocomponent, how it has been working on a particular disease, what is the target protein, how it has been working, what is the mechanism of action, and what are the toxicity effects, what is the maximum dosage which it will give a beneficial result. So all these, they have been developing with respect to the advancements in the research. Can we go to the next slide, please? No, the previous one, sir. Previous one. Yes, this one. So if you can see here, the definition of pharmacognosy. Etymologically, it means the knowledge of poisons or remedies. So when we take any substance, even if we take food, even if we take sugar in a lot many quantities, then it will be toxic to our body. Similarly, phytocomponents also have the same property. When it is taken in a smaller dose, this will have some therapeutical property. Now, later on, from this definition, there is an advancement to the next level. Now, in this advancement, initially what happened is the isolation of phytomolecules, the traditional system of systematic pharmacognostic study is there. So the traditional system of pharmacognosy is involving with the identification of the plant material and literature survey of the plant material, followed by the extraction methods. After various extraction methods, we'll be going for in vitro activity. Later, after doing that in vitro activity, we'll be going for the isolation of the component from the column chromatography. So, and after isolation of column chromatography, individual components from column chromatography, now we will be going for the fractionation, characterization, and again the in vitro analysis. Now, this process is called as bioactivity guided fractionation. Right. So bioactivity guided fractionation is initially for the extract, we will be checking the activity, and after finding a proper activity along with the general chemical identification test, we'll go for the individual isolation of pure components. Now, when the pure component is present, which can be confirmed by the TLC or any other spectroscopical techniques are there, once after confirmation of the pure component, then we will go for the again activity in specifically. Now, after in vitro studies, we'll go for in vivo studies. We have to correlate the IV IVC. So this is the schematic representation. This is the traditional schematic representation. Now, when we talk about the advancement, the first and foremost one is our reverse pharmacognosy. Can we go to the next slide, please? Yes. So we can see here on your left hand side, you can see like how pharmacognosy acts as a principal and core branch interrelating to every discipline, starting from pharmacology, biochemistry, pathology, botany, whatnot, physics, analysis, chemistry, everything. It is the core discipline which is relevant to all the aspects. Now, moving towards the right side of the screen, you can see one side we will be having a systematic and traditional schematic representation of isolation of a component and its evaluation. On the right side, you can see the reverse pharmacognosy. Now here, reverse pharmacognosy is the first step in the advancement. So what is reverse pharmacognosy? So traditional system involves the selection of the material first and isolation of the compound and in vitro activity, in vivo correlation, like that. But here, if we talk about the reverse pharmacognosy, in this one, we will start with the database. We'll start with the lead molecules. So these lead molecules, these are obtained in a various databases. Libraries will be there, various databases will be there. From there, we'll be finding out some lead molecules. Starting from the lead molecule, we'll be incorporating that lead molecule to one database. Cellinergy is one database, wherein in Cellinergy, we will be having the identification of the target proteins and everything. So in this Cellinergy database, when the molecule has been incorporated, we will be having the doc scoring. So taking the doc scoring, the hits and the leads will go for ADMET analysis, and then for in silico studies will go, and later on in vitro evaluation, later on ethnobotanical database management will go for ethnobotanical ETMDB. We call that one as ETMDB, ethnobotanical database. So this ethnobotanical database, this gives the information. If you keep one molecule in that ethnobotanical database, then we'll be having the entire history, starting from the name of the drug, the biological source, geographical data, its chemical components, its uses, everything will be there. Now, from this ETMDB, we will be selecting the geographical availability. And we'll go to that particular place where there will be diverse, variability in the growth of the plant and all. We'll collect the material, we'll go for the normal process. Now, what is the advantage of using this reverse pharmacognosy? So the advantage here over is, so many a times what happens is our natural products, if you see, our plant material, if you see, it is not combined of a single component. It is a mixture of some hundreds of components together. Now, isolation of each and every component and checking the activity of each and every component is actually a very, a time-taking process, time-consuming process. Now, in order to minimize the time and in order to be very specific with the activity and all, we will be choosing this databases and all. So, first comes your reverse pharmacognosy. I think this concept is clear to all of you. If it is, you can go to the next slide, please. Yes. So if you can just just make a zoom of the slide, I can explain this slide. So in this one, we are having two images. The bottom image where you can see from the molecular level, the genomic level, and till the the organism level. Now here we can say that nature is combined with various we can say affinities. Like from the nature, we are going to have the synthesis of primary metabolites, and from primary metabolites, we are going to have the secondary metabolite formation. Now, taking into a deeper image, these secondary metabolites are formed within the cellular level. And also, one more important thing here we need to consider is depending upon the type of secondary metabolite, we will be having different cycles. The biochemical processes will be varying. Now, here, if you see at the genomic level, right, at the genomic level, each and every organism, suppose microorganisms, if you see, depending upon their genetic information and the genomic code, the toxins or the metabolites they do produce will be varying. So that gives the complexity of the system and their ability and affinity towards the nature. Next, if you go to the first image, so in this first image, I have given 4 sections. Towards the right one, towards the right corner, if you see, that is the holistic approach, experimental as well as holistic approach. So this experimental and holistic approach is the first and foremost experimental section which happened to be done in our pharmacognosy streamline. So if you go for that, that we can say as a traditional medicines. If you see Ayurveda, so when we talk about Ayurveda, they directly use some plant herbs and all directly. In the traditional systems of medicine, our ancient ancestors, they used the directly the parts of a plant. So these parts of a plant, in order to minimize the toxic principles, generally, if we directly go for a natural herbal medicine without proper standardization, there comes the toxicity issues. Now, in order to minimize the toxicity issues, then during our traditional time itself, our ancestors, what they did is they have minimized the toxicity errors by using various methodologies. Suppose if you talk about bhasmas, right? If we talk about bhasmas, which are a traditional system of medicine. Now, the preparation of bhasmas involves heating a particular drug material along with, in the form of a juice or a decoction along with a metallic substance, and we'll be using some sort of fruits during the preparation of asava and arishta. So, asava and arishta actually they are nothing but the fermented products. So this asava and arishta, during the fermentation, they will be releasing some sort of toxic metabolites which are neutralized by a flower. Right. So, like we don't know exactly what the mechanism is, how the toxicity has been neutralized during the traditional system of medicinal preparations, but that comes as a holistic approach. So, if why it is called as holistic approach means homeopathy. So, if you talk about homeopathy, unlike Ayurveda, which the principle is completely different, if you go for homeopathy, here we'll be talking about the principle of law of similars. Law of similars, that is nothing but your like cures like. So here the first and foremost identification of this statement, the law of similars, is given by Samuel Hahnemann. So he performed the test on Syncona bar. So generally what we know is Syncona is used as an anti-malarial, which is used to reduce the fever, the symptoms of fever and all. Now what happened is Samuel Hahnemann, what he did is he found like this drug is going to reduce fever, but it is also having some symptoms which causes fever. Now he has done the same test on a healthy individual. So here this is called as a holistic approach. So in holistic approach, we will be taking a group of individuals who are healthy and who are not healthy. So this practice we were discussing now in the clinical trials, the different phases of clinical trials. But in when these systems are not have been developed, then in the ancient traditional systems only, our ancestors, Samuel Hahnemann, he has described about this holistic approach, treating or utilization of the drug in a healthy volunteer as well as a person who has the same side effects or the same ailment, we can say. Right? So this is the first one. And going to the second one, if you see, that is the next advancement. This second slide, going to the next, you can say part section B, the green colored, half green colored one, the left lower side. So here, the example for this one I can suggest is our bioassay guided fractionation. So in this is the next level advancement from the traditional approach or the holistic approach. The next one is the next level advancement wherein we'll be directly using isolating the extracting the active component, and from the active component we will be isolating the individual pure components. So this comes as bioassay guided or bioactivity guided fractionation, wherein first of all plant has to be selected, extract has to be extracted out. Later from the extract, individual phytocomponents have to be isolated, and those phytocomponents have to be checked for their activity on animal models, in vitro models as well as animal models. So this is the second level of advancement. So the third level of advancement, if you see, so in the third level of advancement, as we have earlier, just before I have discussed about the reverse pharmacognosy, wherein we have been talking about the library databases. So this library databases, they will be containing a lot of information regarding the plant molecules or the phytocomponents or the protein targets or whatever it is, right? So the third level of advancement is our utilization of computer-aided drug design, wherein first of all we'll go for in silico studies. Depending upon the doc score of the in silico studies, depending upon the ADMET analysis, then we'll go for the in vivo correlation, in vitro correlation as well as the in vivo correlation. That comes as the third advancement. And the last one, if you see, again that comes as a holistic approach, wherein first of all in silico approach, in vitro approach, in vivo approach, then going for clinical trials. Here, the last one, again coming to the holistic approach. So this is the basic line where our advancement has been starting from the traditional systems of medicine. Okay, we can go to the next slide. No, the previous one, yes. Okay, so in this one, if you see, no sir, next one. Yeah, so if you can see here, we can talk about the sources of the drugs, which are, I told you about the different databases like the phytochemical databases, phytocompound databases will be there, genomic databases will be there, protein databases will be there, and also ethnobotanical databases will be there. So depending, we will be using in pharmacognosy nowadays, we are using all these types of databases for analyzing one drug into a new discovery for a particular ailment. Now here comes one more advancement, that is our drug repurposing. So generally, if we talk about one, quinine, Syncona, we can take an example. So Syncona, it will be containing two active ingredients, mainly acting two active ingredients, that is quinine and quinidine. So quinine and quinidine, these are stereoisomers. The whole structure is same. They are isomers to each other. We can say enantiomers to each other. Just with this simple isomerism, there is a lot of difference in the activity. So quinine, this is particularly used in the treatment of malaria, anti-malarial activity, and quinidine, just because of the isomeric nature, it is used as arrhythmic. The same thing happens with this one. What can say? Morphine. Morphine, if you see. So morphine, this is used in the treatment of, post-operative pain and all. right? So this is used as a high-class high-class analgesic. Now, the derivatives of morphine are thebaine and codeine. So codeine, this is used as antitussive. Now, if you talk about codeine, this is nothing but same morphine, which is methylated. Methyl morphine is nothing but our codeine. So this is one example we can cite under drug repurposing, wherein by altering the structural change or else by altering the functional groups in a particular moiety, we can expect a change in the activity. Right? This is the another advancement, drug repurposing. Okay. Yes. Next, we can go to the next slide. Yes. Now, talking about the innovations in digital pharmacognosy. Right. So, innovation, digitalization is not only relevant only to pharmacognosy, it is integrated with the humankind only, the whole humankind only. So, initially, when we want to talk, we used to have a simple mobile. That is the only mode of communication. Later, text, SMS messages. Later, Orkut is there, WhatsApp is there. This is digitalization. Now we are having AI. For everything, we are having AI, artificial intelligence, right? So, in the same way, this AI has been overtaking everyone, every humankind, human brain with respect to the drug discovery. Now, what is this digitalization with respect to the pharmacognosy? Integration of digital technologies in the drug discovery. This is nothing but our digitalization in pharmacognosy. So, next slide, please. So in this digitalization, we will be mostly talking about the in silico studies or various databases which we are having. Okay, yes, we can go to the next slide, please. Yes. So in these innovations in digital pharmacognosy, we can talk about various innovations, like we can talk about high throughput screening, we can say, or else we can talk about metabolomics, chemoinformatics, bioinformatics, or else AI and machine learning, blockchain technology. So all these are there in our innovations in digital pharmacognosy. So if we go one by one, first of all, if we have to start by, first of all, let us start with CAT. So CAD is nothing but computer-aided drug design. So computer-aided drug, there is a very great difference between the normal research, wet lab and the dry lab. So wet lab means the traditional pharmacognostic research, the traditional research wherein we'll be using all the amenities in the lab, starting from the scratch till the final output. But if we talk about this dry labs, right, like the in silico studies, CAD and all, we will be going on with the assistance of digital technologies. We'll be taking the help of computer-aided drug designing, wherein we will be finding out the receptor. Suppose if we talk about the resistance of antibiotics, so many antibiotics are there. Initially, penicillin is there. Penicillin has been replaced by methicillin, right? So the difference is just because to overcome the resistance. So penicillin, this generally acts by 5 different mechanisms. First one is the cell wall inhibition, like cell membrane breakdown and all, right? Inhibition of cell membrane breakdown and all. Now here, how we are going to initially in olden days, it became a very tough task for changing one drug to another drug resistance. Like this became a very huge problem for tuberculosis or any other diseases. Right? But nowadays it became so easy that because of digitalization, we can go with the protein ligand binding. In silico design, we can do. And with the help of our target molecule, we can do the binding studies. And we can go with the DOC scores. We can go with the ADMET studies like absorption, distribution, metabolism, elimination, and toxicity studies. That is ADMET analysis, as well as pharmacokinetics also, drug metabolism and pharmacokinetics. So this ADMET analysis as well as drug metabolism and pharmacokinetic studies, it became very easy to overcome all these steps, and with a simple DOC score, which we bind with the protein as well as our target molecule, we can directly go with various formulations, various combinations of drugs. So these combinations of drugs not only give us the DOC score, also one more important thing is they can also give our toxicity profile. They can also show us various genetical parameters which we can change in order to go with the drug repurposing and all. Right? We can go to the next slide, please. Yes, we can skip to the next slide. Yes. So if we go with the AI and this one, AI and machine learning. So here we can talk about the artificial intelligence and machine learning. So in this artificial intelligence and machine learning, we can optimize various extraction processes. We can go with the drug profiling. We can go with the ADMET analysis. How this is possible? How all these analysis, prediction of targets, how this is even possible means this is possible with the help of various databases. Okay, so what are the various databases we'll be using? So if, please go to the next slide. Yes, so here you can talk about, I have cited one example, DeepMind's AlphaFold database for protein binding and all. So here if you see anti-cancer drugs. So anti-cancer drugs are like nowadays it is very difficult to treat the cancer. Right. So the main problem here is because of the genomic variation. So cancer is nothing but disease, or we can say an ailment which occurs because of the variations in the genetic hindrance or the genetic quality of the particular cell. Now, if the genetic information has been completely altered, a new cell which encodes for a different DNA, it will be forming, and which is not at all relevant to the our human body. So that is the reason why it grows in our body, which is not even being diagnosed at the early stages. Now, once after it reaches to the third stage or fourth stage, that is the last stage where it will be traveling to the whole body. And like when it gets traveled into the bloodstream, because of the porosity, like the cell membrane porosity, which is very thin, it reaches each and every body in, I mean, every part of our body. Now, here what happens is when it passes through the bloodstream and reaches every part of the body, now the encoded gene, which is very different to the normal genomic information of our body, this is coding for the formation of a new cell. So when this is coding for the formation of a new cell, there comes the replication of DNA, replication of the cells. One cell is giving rise to number of cells forming a tissue which was coming, I mean, diagnosed as a cancer. Right. Now, in order to minimize this cancer, first of all, we have to, I mean, if it is a traditional approach, we have to go for the extraction, isolation, characterization, all these things we have to go, which takes a lot of time. But when we talk about this digitalization, here we will be targeting the protein binding, right? The protein and the receptor binding. So when the protein and receptor binding has been targeted, like in during the in silico studies or the, we can say, CAD studies, right? We can give the toxicity, how much toxicity the genome is causing, whether it can be removed or it is benign or it is like not benign, malignant and all. Can go to the next slide, please. So in this one, this is a representation of whatever the like alpha mines, protein binding and all. So here, if you see in this image, no, yes, here if you see in this image, so we can talk about the analysis. Yes, on the right hand side, if you see the analysis, whatever the blue colored proteinous structures were there, like whatever the blue colored thread like structure is there in the protein, that is considered as benign, and the red color is indicating the pathogenicity or the malignancy of a particular antigen. Right. So here we were discussing about the antigen P53. You can see antigen P53 binding target. Okay, the next one is high throughput screening. So high throughput screening here, this is also known as HTS or VHTS. So high throughput screening, this is a library. The next slide, please. Yes. So high throughput screening is a database wherein we can rapidly screen the molecules, the phytomolecules with respect to their biological activity. So this in this database, we can have the nature of the smallest phytomolecule and its activity, its biological activity, and its target proteins, everything. We can have everything in this HTS database. Now here we are having two types of HTS database. One is normal high throughput screening databases, and the second one is virtual high throughput screening database. The only difference between these two is in high throughput screening, we will be utilizing just the in vitro methods, like in the database only, we will be having the protein target binding, the availability of the molecule, its dog score, everything will be available in vitro only. And finally, we'll go with the in vivo studies. When we come to this virtual HTS. Virtual HTS is nothing but virtual high throughput screening method. So in this virtual high throughput screening methods, we will be integrating this one with the available CAD or the in silico drug design. So we will be utilizing this in silico studies along with the in vitro analysis as well as the in vivo analysis. This is the high throughput screening. And yes, the next one. Yes, the next one is bioinformatics and computational biology. So in this bioinformatics and computational biology, we can go to the genomic level. So starting from the genomic information, like here we can use the tools like BLAST as well as PyMol. So PyMol, this gives the information regarding the smallest molecule and its ability with the genomic sequencing. And if we talk about this BLAST, so this gives the alignment. So BLAST gives the alignment as well as a sequencing of a protein. So here, what the main advantage of this bioinformatics and biology is we can get the data of the genomic sequence as well as the DNA sequencing, RNA sequencing, everything we can get. So that once we have the data of the genomic sequence, we can have the multiple alterations. So we can have whether this genomic structure has been altered. Can this be able to overcome the resistance offered? Can this be able to give rise to a new metabolite like that? So each and every secondary metabolite is coated by a particular cell which has encoded with a particular genetic information. Now, like how we are going with the hybridization techniques wherein the genetic information has been changed, the mutation where the genetic information has been changed, like in plant tissue culture and all. Likewise, here also, we will be going in depth till the genomic level. So in the genomic level, the researchers will be researching on the alteration, the possible sequence aligning, wherein we can go with the formation of a new protein molecule or a new sequencing of a DNA or RNA, which will encode for a new metabolite like that. So this help. You can go to the next slide, please. Yes, you can see here the right side screen, it is giving you the structure of a protein, right? PyMol, that is a database, PyMol database. And the left side, if you see, that is the BLAST database, wherein that gives the alignment of the genomic sequence, right? So with the alteration, with the alignment sequencing of the genomes, the genetic information of the DNA and the RNA, or else the protein binding affinity and all, we can code for a new novel lead molecules. So here we will be talking about hits and leads. Okay, so first of all, when we utilize all these techniques, let it be HTS, AI and machine learning, bioinformatics and chemoinformatics, metabolomics, everything, when we use all these digitalization techniques, we will result in the lead molecules. So this lead molecules, after data mining, a very serious data mining, this will result in the formation of a hit molecule. So these hit molecules will undergo the clinical trials and all. So once after completing the clinical trials, this will be releasing into the market as a formulation. Next slide, please. Yes. So in this one, metabolomics and chemoinformatics. So in this metabolomics and chemoinformatics, again, starting from the basic metabolite, starting from the cellular level. Suppose, let us talk about the pathways. So here, if you talk about the reserpine, right? Like let us take the example of reserpine. So reserpine, this comes under indole alkaloids. So this contains the indole nuclei. Indole nuclei is the origin of indole nuclei is from the amino acid called as tryptophan. So tryptophan is obtained from the shikimic acid pathway. Now, in the shikimic acid pathway, erythrose 4-phosphate is the starting material, and erythrose 4-phosphate is formed from glucose molecule, and glucose is obtained by the photosynthesis. Now, photosynthesis is obtained from the entire process involves carbon dioxide, wherein if we want to check the whole process starting from the scratch, starting from the minimum metabolite, we want to check the whole process. Now we will be going to the basic level. Now we want to like we want to go for a novel isolation of a novel metabolite during the shikimic acid pathway, and we have done that by in silico studies. Let us assume that we have done we know the shikimic acid pathway very well, and in the shikimic acid pathway, like we are assuming, okay, fine, if the structure of chorismic acid has been altered, there might be a change in the formation of a newer metabolite. Let us see whether this can be done or not. Now, in this case, what we'll be doing is we will go to the scratch level. The scratch level is nothing but your formation of glucose. Now, how the glucose is formed? It is formed by the photosynthesis. Now, we cannot do anything to the photosynthesis. We cannot change the photosynthesis process. Now, what we'll be doing is the plant in which we are going to check the shikimic acid pathway, this will be exposed with the radio-labeled substance. This is considered as a biomarker. So this radio-labeled carbon dioxide, we will be exposing this plant material to the radio-labeled carbon dioxide. So this radio-labeled carbon, which is there, 13C, which is there, this will be present throughout the whole cycle. Now, when the glucose has been formed, this will be undergoing the glycolysis process. It will come to erythrose 4-phosphate. From erythrose 4-phosphate, this will go to shikimic acid, shikimate phosphate, next chorismic acid, next again anthranoic acid. All these amino acids are formed. Now, if I want to alter any structural change, now first of all, I'll go with the in silico studies. Once doing the in silico studies, I will get an idea whether this change has been resulting in any useful molecule or not, and how we have to go for the identification of whether the molecule is useful or not is by doing the ADMET analysis, by doing the toxicity studies, by doing the doc scores and all. Now, once everything is done, like the ADMET analysis is done, doc scoring is done, now we will do the same process with the in vitro analysis as well as the in vivo analysis. Later, the same thing we can do with the correlation of IVC studies with the natural environment. So this is how metabolics is designed. Metabolics is mainly concerned with the individual study of a single metabolite from which the final metabolite is formed. Right. The next one is chemoinformatics. So chemoinformatics, here we'll be having different databases for metabolites. Metabolites is one database for metabolomics, as well as human metabolite database. HMDB is human metabolite database. So this human metabolite database is another database which gives the proper information about the metabolomics. Now, talking about the chemoinformatics. Now, this chemoinformatics, we are having different databases for this chemoinformatics as well, like the Metrabase and all. So in this chemoinformatics, the database will be constituting the information, each and every information, starting from the molecule, the structure of the phytocomponent, the molecular formula of the phytocomponent, the structural, like SAR, structure activity relationship of the particular phytocomponent, the spectral data, each and everything was incorporated in the Metrabase of, like, that is a database of chemoinformatics, right? The next one. Next slide, please. Yes. So here comes the next slide. So here I have represented the chemoinformatics, wherein you can see here the phytocomponents. The yellow colored one is the phytocomponents which are having the minute details starting from the molecular formula, molecular structure, the doc scorings, not the doc scorings, molecular formula, molecular structures, the SARs, as well as the spectral data, the uses, the pharmacological uses, everything was incorporated or encrypted in the chemistry space, that is the database of chemoinformatics. Now, if you go with the this database, taking this database, we can go with the ADMET analysis, and we can go with the protein binding studies like in silico studies, and followed by the in vitro studies and in vivo studies. And when we come up with a good doc score, we'll go with the in vitro studies. And when we again have a very good result with the in vitro studies, we'll go with the in vitro in vivo analysis, wherein we'll have a resultant with respect to the diseases and all. Please go to the next slide. Yes. So this one is the blockchain technology. So in this blockchain technology, here this is the very important, we can say very important part of the digitalization, wherein Here, the procurement, starting from the origin of the raw material, where it is procured from, how it is procured from, and during the processing, what are the various steps we are taking. Like we can say recording. We can say as a data piling up. So this blockchain technology is the software which will incorporate all the minor data from starting from the procurement of the original material, original raw material, till the processing and the release of the final pharmaceutical dosage form. This is given in the blockchain technology, right? So these are the some of the we can say advances or digitalizations in pharmacognosy specialization, which will ultimately result to the drug analysis and drug discovery. Now, talking about the next one, we were talking about the key challenges. Yes, we have enough data and enough databases for storing all the relevant data, novel molecules. We have phytocomponents, we have protein database, we have genomic databases, we have, and like the chemistry, like the chemical databases, we have everything we are having. But we are also having some challenges with respect to all this digitalization process, like how encryption is necessary for our regular utilization of mobile. So encryption is also needed for this digitalization in our drug discovery process, wherein the key challenges involve the data integration and management and the standardization process, right? We can go to the next slide, please. Next slide, please. Yes, data integration and management. Yes. These are the key challenges. So standardization issues, we can talk about validation, ethical issues. So all these are various key challenges, how we have to cope up very carefully with respect to the digitalization. Now, talking about each and every one, data integration and management. If you talk about the data integration and management, now this data integration and management really makes a sense in the way how we have to store the data without any possible, we can say, hacking and all, right? Without any possible hacking or rewriting of the databases and all. Suppose if we take this Wikipedia. So Wikipedia is one database where we can have the access to each and everything. Like if I search about pharmacognosy, it will give the whole data about pharmacognosy. Now, if I want to change something, I can change. I have the access to change. So here what happens? Without a proper validity and proper proof, we are incorporating some information into the database. So in such case, there is no standardization, right? So this leads to misinformation, like misguiding. Okay, so the first one is data integration and management, wherein we have to we have to talk about the security of the databases, right? Initially, in the Metabolomics, I have discussed about two types of databases. The first one is Metabolites, and the second one, we can say it is human metabolite database. So these two databases, these are maintained in such a way that the basic information, starting from the cellular information, the molecular level information, it has been encrypted in such a way that only after having a valid proof, this is utilized. And how this has been utilized is in the discovery of a novel drug components. So novel drug components in the sense, many ailments, like we can say cancer, we can say, or else we can also talk about some viral infections. We can talk about the resistance of microorganisms. So basing upon this databases, the information which has been protected and integrated in this databases, which is encrypted in this databases, we will be changing and altering some structural changes in a molecule or a protein and the target receptor, thereby leading to the formation of a new drug molecule, which is a lead molecule. And this lead molecule will give rise to the hit molecule. Right? Yes, go to the next slide, please. Yes, data integration and management. The next one. Next one is standardization. Yes, in the standardization, as we all know, standardization, why we have to do the standardization? So we need to give the results accurately. We need to reproduce the same results, right? So this reproducibility of the results is very much important. Why it is important means, in order to go for a discovery of a new molecule, if the results have been altering always, if the results have been altering, that leads to again a misguided information. which is not properly designed, right? So again, standardization has to be done. Again, we are also having some databases wherein the information is standardized. The standardized information has been encrypted properly. So the information in this standardized databases has been utilized again in the drug discovery process. Yes, the next one. Interdisciplinary collaboration. Now, this is a very interesting aspect. Interdisciplinary collaboration is, we can take the example of National Institute of Health. So this National Institute of Health, this is mainly targeting the microbiota, human microbiota. So what they are doing is they are utilizing the natural phytomolecules. The natural phytomolecules means they are not giving the natural phytomolecules in the form of a drug molecule. They are simply like, if you talk about this United States and all the European countries, unlike the South Asian countries like India and all, if we talk about the European countries and so all the European countries, when a person goes for diagnosis or any treatment to a doctor, the doctor prescribes the medicine, right? So along with the prescription of a medicine, there will be always the assistance of a dietitian. Now, this dietitian, he or she, they will be allocating some what we can say the foods or the food items which are naturally consumed. Now, what is the utilization of this food? How the food is giving, we can say how the food is making sense along with the medicine. The thing here is the food which they are prescribed by the dietitians, they will be containing certain we can say certain antioxidant molecules, or we can say any polyphenolic substances or flavonoidal components, which are naturally phytoactive materials. So these natural phytoactive materials, as such, they have synergistic potential. First one is they have synergistic potential. The second one is they have natural ability to clear the toxic cells. I mean, like free radicals, if you see. So free radicals are the major, we can say free radicals are the main important reason for almost all the ailments in our body, starting from pain or inflammation till we can say diabetes or cancer, hypertension, whatnot, everything. It is associated with the formation of a free radical. Now, green tea, especially we are taking green tea nowadays. Now, this green tea, this contains enormous numbers of polyphenols. So these polyphenols, they have the ability to scavenge the free radicals. Now, also, this is one aspect. Second aspect is the synergistic potential. Now, synergistic potential in the sense, some medications, they will not be able to get absorbed by our body. Like our body cannot be absorbing all the 100% of the drug. So when we take 100 mg of the drug, not 100 mg of the drug, paracetamol will be taken up by the body. Only some 20 mg or 30 mg, depending upon the person's ability, the body of the person, the genomic nature of the person, everything matters. So the absorption pattern will be varied. Now, if you say some BCS class 2 drugs, right? BCS class 2 drugs are there, which are mostly in the polyphenolic substances. Most of the BCS class 2 naturally occurring phytochemicals, they come under class 2 substances only. So this class 2 substances which are there, they have poor bioavailability, right? Lesser absorption, poor bioavailability. Now, in order to enhance this bioavailability, we need to give a synergistic potential. So for giving a synergistic potential, again, if we prescribe another medicine, that will be a load on the renal system and the hepatic system. So instead of that, if we give a natural substance, if we give a naturally occurring phytocomponent, which has the ability to scavenge the free radicals and which also has the ability to potentiate the activity of the class 2 substance. Now, this is going to enhance the activity of the medication, right? So this is what nowadays the research is concerned on. Most research is concerned on this one only. Now, talking about this interdisciplinary collaboration, researchers of NIH, National Institute of Health Sciences, they were mostly focused with the gut microbiota of the human kind. Now, humankind means starting from children, teenagers, adults, geriatric patients also, right? So all the persons, they have taken a different age group of persons, different age group of healthy volunteers, and they have been, depending upon the food habits, depending upon the diversity, depending upon the ecology, their genomic pattern, Depending upon all these qualities, they are analyzing the human gut microbiota. Now, how this microbiota has been changing its nature depending upon the all these factors which I have just told you, the diversity, ecology, genomic pattern, their food habits, all these things. Now, if we talk about a particular disease, let us say inflammatory bowel disease, right? Inflammatory bowel disease, like NIH is collaborating with multidisciplinary things like cancer. They are focusing on reproductive sciences. They are focusing on digestive sciences. They are focusing on and isolation of microbial metabolites. They are focusing on. So many things they are focusing on. Let us take one simple example. So in this simple example, I will try to give you about the this inflammatory bowel disease. So we all know what is inflammatory bowel disease. So this inflammatory bowel disease, this has no limitation that this will come only to the geriatric patient or a person who is having high content of spicy foods and all. It comes from different types of food habits. It comes from different genomic patterns, different ecological and cultural diversities and all. Now, these NIH researchers, they are collaborating with this gut microbiota and its ability to overcome this inflammatory bowel disease in all the four different people of different regions, different genomic patterns and all. The same gut microbiota they are taking, they are altering the genomic sequence and they are checking its activity on the volunteers. So this is called as interdisciplinary collaboration. Right? The next one is validation and verification. So in this validation and verification, and the next one is ethical issues. So if we talk about validation and verification as well as these ethical issues, legal and ethical issues as well. Now this both of these two are interrelated in the sense that the standardization has to be done. After standardization, cross-validation or cross-checking has to be done whether it is standardized or not. Next one is the legal issues. Now, talking about the legal issues, we need to talk about the intellectual property rights. So, in this intellectual property rights and data as well as the privacy concerns as well as this legal frameworks, all these things matters over here. Now, we all know that we have been India is India is the home for so many phytochemicals which are unexplored till date. Right. But what happens if we talk about curcumin? Curcumin, it has a lot of potency. We have discovered the potential of curcumin, but patent is not with Indians. It is with other countries. It is with the Americans. The patent right has been taken by the Americans, right? So work has been done here. The material has been procured here. Everything has done here, but the patent has been obtained by the other country persons, right? Here, this is the main important factor we need to talk about. When we talk about the novel drug discovery, with the incorporation and utilization of various digitalization techniques, let it be HTS, let it be VHTS, or else your metabolomics or chemoinformatics, bioinformatics, or AI and machine learning, if we are going to design a novel drug molecule, that has to be patented. We need to take our intellectual property right. We need to follow our intellectual property right. Right. So the data it should be very secured very properly, that it hasn't released till the final what we can say the formulation, till the final formulation has been released into the market, till the patent right has been procured, there should be a secrecy, there should be a hindrance or a border between what is the process that is going on, right? Yes, I think we are done with this. Yes. So this is about the digitalization in pharmacognosy. Uh, you're open to if you have any questions, please. So these are all the references which I have incorporated because this is nowadays this is the technique which is in trend nowadays. I have tried my maximum, and this is what I have got. But still, if you have any doubts, I'm happy to clarify.
Yes, ma'am. It was a wonderful session. The presentation was awesome. So I request all the participants, if you have any queries, you can just post in the comment section. Any queries, you can post in the comment section. Ma'am, we have one query. Can we replace the DNA molecule by using these advanced techniques, or we can inhibit the cell division?
Yes. So replacement of DNA cannot be done. Rather, we can change the sequencing. We can just change the genomic sequencing, right? Replacing of DNA cannot be done.
Any more queries?
Okay, then I think
I think there are no more queries, ma'am. I just want to know, like, if we want to do any in silico studies. Yes, ma'am. So, in what
yes,
stage it has to be done?
means in silico studies we want to do.
Yes, if we want to do a extract preparation, we are doing in the starting, we are taking the raw materials and we are doing the extraction process. Now we need to
Hmm.
do the biological activity, right, ma'am?
Yes,
So, in what stage is the in silico studies has to be done, and what is the importance of doing that in silico studies?
yes. So with the help of in silico studies, we can do drug repurposing. Drug repurposing in the sense like one drug molecule is having one utilization, right? Suppose I told you the example of morphine. Morphine is used as preoperative and postoperative analgesic. Like it is used in the treatment of pain and all postoperative pain. Now, this morphine, when it is like added with a methyl group, methyl morphine, the entire property has been changed. It is used as antitussive now. Okay, so in silico studies, it helps in designing a molecule and finding the binding affinity in silico, right? Computer aided drug designing or the in silico, we can say. Now we know the structure of a molecule, we know the receptor, we know the target molecule. Now, when we go for binding of this target molecule with the protein, we can have the doc scores and all. Now, basing upon this doc scores and all, we know how to how much extent it has been showing its effective therapeutical efficacy and all. Basing on that, we can go for the further uh, proceedings.
We have another query, ma'am. Are there any certification courses to study more about the databases you have explained?
Certificate courses, though, we have computer-aided drug designing till now. I mean, chemoinformatics is also available. Bioinformatics is also available. Certificate courses are available. AI and machine learning, though, I don't think so. It is available now. Yes, there are there.
Thank you, ma'am, for comparing the ancient techniques as well as the modern advanced techniques. It was a wonderful session. I request Mr. M. Ganesh Kumar with proceed with the further session.
Thank you so much, Satya Madam, for a
Thank
wonderful
you, sir.
session. And I would thankful to the management for accepting our request for the conducting of FDP program. And I also thankful to the our principal, Srinivasarao College of Pharmacy, Dr. K. Achyut Kumar sir, for supporting us for smooth running of FDP program. Thank you all.
Thank you
Thank you,
very
madam.
much to all the management of Srinivasarao College of Pharmacy, Principal Sir Dr. Achyut Kumar Sir, Convener, Co-Convener Dr. Swati Priya Ma'am, and remaining all the faculties. I am very grateful for you for inviting me and my session. I hope it will be useful and informative for all of you. Thank you very much. Can I log out of the session, ma'am?